Hello students, I hope you all are fine and doing great. I, Pooja Verma, welcome you all in the second session of chapter 9, that is Biomolecules. The learning objectives of today's session are to help students learn about primary and secondary metabolites, to help you understand about acid insoluble fraction, to make you realize the importance of proteins and the diverse functions performed by them, to help you understand the 4 different levels of protein packaging, to help you understand how polysaccharides are formed from monomeric units of carbohydrates, to develop scientific attitude, creativity, aesthetic sense and problem solving approach in you, to enable you to find the applications of the concepts in your daily life. If one were to make a list of biomolecules, such a list would have thousands of organic compounds including amino acids, sugars, etc. These biomolecules can also be called metabolites. Some of these metabolites like carbohydrates, fats, proteins, nucleic acid are required for their basic metabolic processes and are found throughout the plant kingdom. Even in animal tissues, one notices the presence of all such metabolites. These are called primary metabolites. However, when one analyzes plant, fungal and microbial cells, one would see thousands of compounds other than these called primary metabolites. Example, alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices. These are called secondary metabolites. These secondary metabolites are produced by plants in very small quantity and their extraction from the plants is very difficult as well as expensive. While primary metabolites have identifiable functions and play known roles in normal physiological processes such as photosynthesis, respiration, protein and lipid metabolism, we do not at the moment understand the role or functions of all the secondary metabolites in host organisms. However, many of them are useful to human welfare. Example, rubber, drugs, spices, scents and pigments. Some secondary metabolites have ecological importance. Now let's discuss some examples of secondary metabolites. Plants produce pigments like carotenoids and anthocyanin. These pigments are secondary metabolites. Carotenoids produce bright yellow, red and orange color in various vegetables and fruits like carrot, papaya, mango and pumpkin. They have an important antioxidant function, which means they destroy free radicals and protect us from cancer. Some carotenoids are converted by the body into vitamin A, which is essential for healthy eyes, growth and development. Anthocyanins are water-soluble pigments. They may appear red, purple, blue or black depending upon their pH. Blueberry, black rice, cabbage, cranberries, cherries contain anthocyanins. Plants also produce alkaloids like morphine and codeine, which are secondary metabolites. We still do not know why plants make morphine and codeine, but they are beneficial for humans. Morphine formed by plants is an effective painkiller. It acts directly on the central nervous system to decrease pain. Codeine can also be used to treat severe pain. Plants contain terpenoids like monoterpenes, diterpenes, etc. Terpenoids are the largest group of plant secondary metabolites. These naturally occurring chemical compounds are highly diverse in chemical structure. They are highly beneficial to humans because they have antimicrobial, antifungal, antiviral, anti-inflammatory and antioxidant properties. Plants prepare lemongrass oil because it has a property to repel insects like fleas, ticks, mosquitoes. This lemongrass oil is used by humans as a flavoring agent due to its lemon scent. Plants also produce toxins like abrin and ricin, which are secondary metabolites. Abrin is a biological toxin found in rosary pea plant seeds. The seeds are red with a black spot covering one end. Ricin is a toxin found in the seeds of castor plant. In the body, it releases toxins which inhibit protein synthesis leading to cell death. Plants produce lactins like concanavaline A. Lactins are carbohydrate binding proteins. Concanavaline A is a lactin extracted from jack bean plant, which is a leguminous plant. Students, you are aware that we use turmeric in our daily life. It has been used in India for thousands of years as a spice and as a medicinal herb. Turmeric contains curcumin, which is again a secondary metabolite produced by turmeric plant. Turmeric has strong anti-inflammatory and antioxidant capacity. It has numerous other health benefits. Many of you might have seen periwinkle flowers, commonly called sadabahar in Hindi. Its scientific name is Vinca rosea. You will be surprised to know that it forms a secondary metabolite called vinblastine, which is a very effective chemotherapeutic drug used to treat different types of cancers like lung cancer, brain cancer, testicular cancer, etc. Plants also make polymeric substances like rubber, gums, cellulose, etc. as their secondary metabolites. We use these substances in our daily life. I hope you all remember that while doing chemical analysis of a living tissue, we grind it in trichloroacetic acid, and on filtration we get acid soluble pool and acid insoluble pool. Acid insoluble fraction has 4 types of organic compounds, that is proteins, nucleic acid, polysaccharides and lipids. Except lipids, other molecules have very high molecular weight in the range of 10,000 Daltons and above. The molecules in the acid insoluble fraction except lipid are polymeric substances. Now a question must come to your mind. Why do lipids whose molecular weights do not exceed 800 Dalton come under acid insoluble fraction? Why do they precipitate along with other macromolecules in the acid insoluble fraction? To find an answer to this, recall that lipids are present in structures like cell membrane and other membranes. Whenever we grind a tissue, we are disrupting the cell membrane, due to which the cell membrane is broken into pieces and form vesicles. We all know that lipids are not water soluble. These membrane fragments in the form of vesicles separate along with carbohydrates, proteins and nucleic acid in the macromolecular fraction of acid insoluble pool. With respect to their size, lipids are not strictly macromolecules. Proteins form about 12% of cell content. They are the most abundant macromolecule of the cell. They constitute about 50% of the dry weight. Proteins show an unlimited variety and may have any number of amino acids present in any proportion and arranged in any sequence. Proteins are polymers of amino acids joined together with the help of peptide bond. They help in growth and development and increase muscle mass and strength. 20 types of amino acids occur in proteins and they are called protein amino acids. Plants can synthesize all the amino acids required by them. Animals, however, do not have this ability to form all the amino acids. Certain amino acids cannot be prepared by human body from raw materials. Such amino acids are called essential amino acids. We must obtain them through our diet, otherwise deficiency symptoms will occur. They are 9 in number and include histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine. The remaining 11 amino acids can be synthesized by human body. They are called non-essential amino acids. Now let's discuss some of the important functions performed by proteins. We all know that collagen is an intercellular ground substance. It is also the most abundant protein in animal world. It is the major component of connective tissue that keeps our skin wrinkle-free and bones healthy. All enzymes except ribozymes are made up of proteins. Trypsin, an enzyme which helps in the digestion of protein, is itself made up of proteins. Various hormones are also made up of proteins. You have studied about insulin hormone in your previous class. This hormone is prepared by pancreas, a gland located behind the stomach. Insulin controls the blood glucose level and it is made up of proteins. We all know that white blood cells are the soldiers of human body and they fight against infectious agents like bacteria, viruses, etc. White blood cells make antibodies which belong to globulin class of proteins. So technically speaking, without these globulin proteins, we will not remain healthy as our body won't be able to kill germs. Receptors present in our nose and tongue which help us to detect smell and taste are made up of proteins. GLUT4, the receptor which allows glucose to enter inside the cell, is also made up of proteins. Ribosco is the most abundant protein in the whole biosphere. It is an enzyme made up of proteins used by plants to prepare their food. We will now discuss about the structure of proteins. We have already mentioned that proteins are heteropolymers containing amino acids joined with the help of peptide bond. Biologists study protein structure up to 4 levels: primary, secondary, tertiary and quaternary. Primary structure is the description of how many amino acids are present in the protein chain, which amino acid is the first one, which is the second, and so on. Primary structure depicts the sequence of amino acids present in the protein. As you can see on the screen, conventionally the left end of the protein primary structure is represented by first amino acid called N-terminal amino acid. The right end is represented by the last amino acid and is called C-terminal amino acid. A protein thread does not exist throughout as an extended rigid rod. The thread is folded in the form of a helix, which is similar to a revolving staircase. In proteins, only right-handed helices are obtained. Other regions of the protein thread are folded into other forms in what is called secondary structure. The two major secondary structures are alpha helices and beta structures. In the alpha helix, the polypeptide chain is spirally arranged in right-handed manner. The helix is stabilized by hydrogen bonds between oxygen of carboxyl group of one amino acid residue and amino group of next fourth amino acid residue. Alpha helical coiled secondary structure is found in several proteins such as keratin protein, which is found in our hairs, myosin protein found in the muscles, etc. In the beta pleated secondary structure, two or more polypeptide chains get interconnected by hydrogen bonds. A sheet is produced instead of a fiber or a rod. This type of secondary structure is called beta pleated sheet. Silk fibers contain fibroin protein which contains this structure. Secondary structure undergoes further coiling to form tertiary structure. The long protein chain is also folded upon itself like a hollow woolen ball, giving rise to tertiary structure. It brings amino acids closer which were far apart in the linear sequence. This structure is stabilized by several types of bonds such as hydrogen bond, ionic bond, van der Waals interaction, covalent bond, hydrophobic bonds and disulfide bonds. Quaternary structure is only found in proteins which are made up of more than one subunit. We all know that our blood is red in color due to the presence of hemoglobin found in the RBC. Hemoglobin contains two alpha chains and two beta chains which together constitute the human hemoglobin. Let's now discuss about polysaccharides. They contain monosaccharides as building blocks. In this section, we will discuss about some polysaccharides such as cellulose, starch, glycogen, inulin and chitin, which is a complex polysaccharide. Paper and cotton, which we use in our daily life, are obtained from plants and are made up of cellulose. It is the most abundant organic substance of the biosphere. It is a form of polymer of glucose. Homopolymer means containing only one type of subunit. Successive glucose residues are joined together by beta-1,4 glycosidic bond. Our body does not contain the enzyme which can break the beta bond of cellulose. That is, cellulose is not acted upon by the enzymes present in the human digestive juices. It acts as roughage. Grazing animals like cows and buffaloes can digest cellulose, as the stomach of these ruminants contain microorganisms capable of digesting cellulose. Termites also possess microorganisms in their gut for digesting cellulose. Inulin is a polymer of fructose. It is found in the roots and tubers of dahlia plants. Our body cannot metabolize inulin and is readily filtered through the kidneys. So, can we use inulin for testing kidney functions, especially globular filtration? Yes, we can do, and so inulin is used in testing kidney function. How about starch? You might have done starch testing in your previous classes. Once we add iodine solution on starch, it becomes blue-black in color. Starch has helical secondary structures in which it can hold the iodine molecules, giving blue-black color. Plants store food in the form of starch. Thus, starch is a storage polysaccharide of most plants. Glycogen is a polysaccharide food reserve of animals, bacteria and fungi. It is commonly called animal starch. It is mainly stored inside our liver and muscles. It is made up of glucose. Glucose residues are arranged in a highly branched bush-like chains. As you can see on the screen, in the straight portion, alpha-1,4 glycosidic bond is present. That is, carbon 1 of one glucose joins with fourth carbon atom of next glucose molecule, as you can see on the screen in lower red circle. At branches, alpha-1,6 glycosidic bond is present. First carbon of one unit joins with sixth carbon of next unit. Can you name the molecule which is present in the cell walls of fungi? It is chitin, which is the second most abundant organic substance. It is found in the cell walls of fungi and exoskeleton of arthropods. Chitin is soft as well as leathery. Therefore, it can provide strength as well as elasticity. Its monomeric unit is N-acetyl glucosamine, a nitrogen containing glucose derivative. Thus, it is considered as a complex polysaccharide. Let's have a quick recap of what we have learned today. In today's session, we have discussed about primary and secondary metabolites. We also discussed the reason why lipids are found in the acid insoluble fraction. We have also learned about the importance of proteins and the various functions performed by them. We also discussed about the 4 different structures of protein and the various types of polysaccharides. Before we conclude, here is an assignment for you related to the session. What are the various harmful effects of aspartame, an artificial sweetener in our body? Compare the advantages and disadvantages of consuming jaggery over sugar with respect to human health. You might have noticed in various TV ads about anti-aging creams which claims to have collagen boosters in them. Why do they contain collagen? With the help of well-labeled diagrams, explain 4 different structures of proteins. Hope you all enjoyed the session. Keep smiling, keep learning. Thank you.